Check it out now, y'all.
NanoHub U, online instruction.
If you recall our last lecture, we tried to make the point that solid state of matter contains fundamentally two different types of materials. Those materials that are intrinsically amorphous, and another class of materials which are known as crystalline solids. These crystalline solids basically have a periodic ordered array of atoms that define the structure, the microstructure of a crystalline material. And at the end of last lecture, we tried to make the case that we have to define a unit cell to adequately specify the structure of the crystalline material. And so in this lecture, what we're going to do is we're going to focus on some of the properties of these unit cells, and we're going to discuss a universal notation that's adopted worldwide to specify planes and orientations of planes and directions of lines in crystalline solids. So that's going to be the focus of the lecture today. So there are two important concepts that we have to keep straight. One concept is the concept of a unit cell. So if we have a crystalline solid, what we would like to do is we'd like to be able to define a region of that crystalline solid, which is basically the smallest unit that is required to generate that solid. So for instance, very often the unit cell has the shape of a cube and might resemble an object similar to this toy block. Oftentimes unit cells may not be cubic, they could be rectangular, so this is more of a parallelepiped, right? The dimensions of the different directions are not the same, but the angles between the three directions are all 90 degrees. And then you can get these very strange shaped unit cells, which are really rhombuses, right? The dimensions of the unit cell could be different in different directions. The angles between the different directions of the unit cell are no longer 90 degrees. They're different shapes. So whichever unit cell you have, whatever the shape it has, right, it comprises the sort of the fundamental building block to construct these crystalline solids. So the concept of a unit cell is an important idea that you have to have clear in your mind. Second important concept is the grid system that is used to define the crystalline solid. So the grid system is shown schematically in this slide, and it just indicates how you can translate this unit cell in all three dimensions in order to fill all space and produce a crystalline solid. So these crystalline solids, they do have these periodic arrays of atoms, and in order for you to understand the nature of the crystalline solid, you have to be able to specify where the atoms are, and the way you specify the location of the atoms is by the definition of this crystal lattice, right, which only applies to crystalline solids. It does not apply to amorphous solids or glasses. There are two classifications of the unit cell that I have to mention briefly. Right, the unit cell is either called a primitive unit cell or a conventional unit cell. And so the point I like to make is the definition of the unit cell is somewhat arbitrary. Different scientists in different regions and different labs could define a unit cell for the same crystalline material in different ways. And so it becomes important to define a unique unit cell, and that unique unit cell is referred to as the primitive unit cell. The primitive unit cell tends to have atoms only at the corners of the unit cell, and it's the smallest volume that's required in order to construct the crystalline solid. The conventional unit cell is more a definition of convenience. right? There are different sites within the unit cell where atoms can be positioned, and these additional sites tend to give rise to a more intuitive representation of the unit cell. And so this is referred to as the conventional unit cell. The conventional unit cell oftentimes allows you to see the symmetry of the crystalline structure much more readily than the primitive unit cell, which sometimes is so small that the symmetry of the atoms is obscured. So just to make the point, it's simple enough to do in 2 dimensions. If we have a collection of atoms that are periodic in 2 dimensions, as I show by these dots, I indicate in green some choices for primitive unit cells, right? So these primitive unit cells can be defined by 2 vectors that are indicated in red. The orientation of those 2 vectors is somewhat arbitrary, but you can see that if you, for instance, reverse the direction of this vector, right, you get the equivalent unit cell, and you can see that the unit cell has an atom located at each corner, which defines it to be a primitive unit cell. Somebody else in some other lab could define a unit cell which looks something like this, right? You can see now that the orientation of these 2 vectors has changed, one with respect to the other. The resulting primitive unit cell following this definition is indicated by the green shaded area. An example of a non-primitive or conventional unit cell is indicated down here. It's also possible to recreate this entire crystal structure by translating this cube or this square, which has an atom located in the center. If I translate this square through a repeat distance, I generate all the atoms in the crystal, and this would be an example of a non-primitive unit cell or a conventional unit cell. Unit cells that are not allowed would have a shape that under translation does not allow you to fill all space. So for instance, if someone decided to define a unit cell that looked like this triangle, that would not be valid, because if you translate that unit cell in the XY plane, it's not possible just under translation alone for you to fill all the space that's taken up by the location of atoms. So that's just a simple example to try to convey some of the nuances that go behind the definition of these unit cells and these conventional unit cells. The entire subject of crystallography is a very complicated one, and it's certainly not something that we're going to discuss in any great detail. But basically, it's an exercise in organizing space. And what I try to do here is I try to give you the different steps that are required for you to understand the crystal system and the crystal symmetry of the unit cell. It basically requires you to specify a unit cell. Once that unit cell is specified, then you have to identify all possible rotations, reflections, and inversions that take that unit cell into itself. Surprisingly, these rotations, reflections, and inversions only define 32 possible crystallographic point groups. So of all the crystalline solids that have ever been discovered, there's only 32 possible point groups to describe the sets of rotations, reflections, and inversions that characterize each of those unit cells. These point groups, in turn, give rise to different lattice types, and it turns out there are 4 lattice types. These lattice types are known as primitive, body-centered, face-centered, and base-centered. So these lattice types are something that you need to know, and I'll try to point that out in the next slide. These lattice types basically allow you to define the structure of the unit cell. You then have to translate that unit cell through space to produce the entire crystallographic collection of atoms, and that translation of these lattice types has to fill all space, and that allows you to define space groups that characterize these different translations. From all these possible lattice types and unit cells, you, at the end of the day, find out there are only 14 different space-filling lattices that can be defined, and these 14 space-filling lattices are referred to as Bravais lattices. So Bravais was a crystallographer back in the 1850s who came up with this system. Of these 14 Bravais lattices, you have to then go back and look and see how they're organized in terms of the point groups. Turns out there are 7 distinguishable point groups out of the 14 Bravais lattices, and these 7 distinguishable point groups then give rise to the 7 crystal systems that are universally recognized as possible for specifying crystallographic structures of single crystalline materials. So I put a few slides in the appendix to try to flesh this out a little bit. The bottom line here is you have to know there are 4 different lattice types, and you have to know that there are 7 different crystal systems. That's the output of this really rather complicated program that identifies the various symmetries of the unit cells that are possible. The 7 crystal systems are listed in this slide. What I've done is I've ordered the crystal systems from the least symmetric to the highest symmetric systems. The unit cells that have the lowest symmetry, that's called the triclinic system. There's 1 atom at each corner of the triclinic unit cell. The dimensions of each side of the triclinic unit cell, those are not equal to one another, and the angles between the various directions in the triclinic unit cell are not equal to 90 degrees. So this is referred to as the lowest symmetry of all the crystal systems that have been discovered. The highest symmetry crystal system involves the cubic lattice. The cubic lattice can either be primitive, in which there's 1 atom at the corner of each vertex of the unit cell. It could be a body-centered cubic. So this is an example of the body-centered lattice type, where there's an atom in the middle of the cube and 8 atoms located on the corners. And then there's the face-centered cubic, which involves an atom located in the face of each at the center of the face of each cube, right? So these are referred to as the primitive, the body-centered cubic, and the face-centered cubic structures. So most of the work that we focus on in this course involves the cubic lattice. That's the easiest one to understand. But in principle, you can get crystals that crystallize in any of these 14 Bravais lattices. So there are 14 different lattices. They're grouped into these 7 different crystal systems. All right, that's the bottom line. How do you specify where the atoms are in a crystalline solid? Well, you have to know these translation vectors that define the unit cell. So the simplest example is in 2 dimensions. If I have a 2-dimensional periodic array of atoms, I can define the location of the atoms by these 2 vectors, A1 and A2. And if this is a good choice for a unit cell, then you can specify the location of any other atom in this crystalline solid as an integer times the vector A1 plus an integer times the vector A2. In 3 dimensions, you have to just extend this definition into the third dimension, and so now you've got vectors A1, A2, and A3, which define the position of atoms with respect to some coordinate origin. A1, and A2, and A3 need not be the same. They need not be oriented at right angles to one another, but if you have a simple cubic lattice, they are identical, so A1 equals A2 equals A3, and by giving the integer numbers L1, L2, and L3, you can specify the location of any atom in that crystalline structure. Sometimes it's very easy to get confused when trying to understand a crystalline solid. So there are many websites, and I just give you one here, and I encourage you to look at it. This website allows you to specify different Bravais lattices. It allows you to specify different crystal structures, and then it allows you to visualize the Bravais lattice and the crystal structure as you rotate the object in 3 dimensions. So nowadays it's pretty simple matter to be able to visualize in great detail the placement of atoms in very complicated structures using these canned software routines. It's important for you to know something about the cubic structure, the cubic system. So this is a standard table that you find in many different textbooks. It basically gives you the 3 lattice types: the simple cubic, the body-centered cubic, and the face-centered cubic. It tells you what the volume of the units, the conventional unit cell is, right? In the case of the simple cubic, which is this case here, that's also the primitive unit cell. So these are the conventional unit cell volumes. They're always, they're the same because the dimensions of each side of the unit cell is A. It tells you how many Bravais lattice points there are per unit cell. It tells you the number of primitive cells in a conventional cell, right? So it just breaks down these 3 systems, and it's a very useful table. And if you can understand the entries in most of these, for most of these items in this table, then you've developed an appreciation for the cubic crystallographic system. So it's a useful reference table that I use probably once or twice a month in the course of doing research on various subjects. I'm often asked what determines the structure of a particular crystalline solid, and that's a really good question. It's also a very complicated question, and I attempt to simplify the answer in this diagram here. And what I try to indicate is that the structure of a crystalline solid is really the result of a tug of war between 2 competing factors. One factor is nature wants to pack these atoms as close to one another as possible. There's a tremendous energy advantage if you can push the atoms so that they're very closely ordered. This implies that there's no directional bonding between the atoms. The only thing that determines the final position of the atoms is how close you can push them together, and that's going to be determined in some sense by the atomic orbitals and the radii of the atomic orbitals that surround the nucleus of each atom. So that's one force that determines the structure of crystalline solids. And this force, or this paradigm, wins out for most metals. At the other end, the highly directional bonding between atoms in a solid is the dominant factor that determines the crystallographic structure. This highly directional bonding is basically a quantum mechanic calculation based on the atomic orbitals of the electrons in the solid, and it takes advantage of the symmetry of these various orbitals that we just touched on a little bit when we discussed the hydrogen atom. this highly directional bonding determines the crystallographic structure of elements like silicon and germanium, which tend to be semiconducting, right? They crystallize in structures that are basically determined by the shape of the electron orbitals in the outermost energy shells of these individual atoms. And so then there's a whole lot of materials that trade off these two different mechanisms, and the resulting crystalline structure is what it is based on how much, based on the dominance of either of these two mechanisms. So in a very very brief way, this is why different materials that are different crystalline materials crystallize in different patterns. We need a very general way to specify the orientation of crystallographic planes and directions of specific directions inside these crystalline solids. And the technique that's been developed to name the planes and to name the crystallographic directions is named after Miller, who worked on this problem a long time ago. And basically, crystallographic planes are defined in terms of things called Miller indices. And these Miller indices are just three numbers, H, K, and L. And the ratio of these numbers, H, K, and L, equal the ratio of the intercepts of a particular plane with respect to the crystallographic directions defined by the unit cell. So, for instance, if we want to specify the crystal plane that I've got sketched here in green, what you would have to do is you would have to find the intercept of this crystalline plane with the x-axis, the y-axis, and the z-axis. So for the example I've got drawn here, the crystallographic plane intercepts the x-axis at 3 units, it intercepts the y-axis at 2 units, it intercepts the z-axis at 2 units. These direction vectors A1, A2, and A3 need not be the same, right? So this is a very general system. It just keeps track of how many units along X, Y, and Z the intercepts occur. So the ratio of H to K to L for this case would be the reciprocal of the intercepts. So I would have 1/3, 1/2, and 1/2. And so H, K, and L have to be the smallest integers that have these same ratios. So what you end up doing is you end up finding a common multiplier that turns these fractions 1/3, 1/2, and 1/2 into integers. So for this case, the smallest multiplier would be 6. And so then the Miller indices would then be 2, 3, 3 for this particular plane. The notation that's been developed is that if you enclose these Miller indices in parentheses, that indicates the plane that's sketched here in green. So if you talk about the 2, 3, 3 plane in this particular crystallographic structure, pretty much anybody in the world would understand that this green plane is the plane that you're referring to, because these Miller indices are used everywhere. One of the virtues of the Miller indices is that you can also specify a line perpendicular to the plane by using the same Miller indices. So if you want to draw a line perpendicular to this plane, if you want to specify that line, what you tend to do is you put these Miller indices inside of square brackets. And so Miller indices 2, 3, 3 with surrounded by square brackets indicates a line that's perpendicular to this green plane. And then lastly, a further advantage of these Miller indices are that they allow you to compute the separation between parallel planes of a similar type. So if you want to know the difference in a cubic system, this equation only works for cubic systems. There are similar equations that have been derived for all the different 14 Bravais lattices that I sketched out in a previous slide, but Most of the work we're going to do in this class is related to cubic systems, right? It turns out the separation between the planes labeled by the Miller indices HKL, that can be written as a very simple equation. So if you tell me what the dimensions of the unit cell are, that's A, and you tell me the Miller indices HK and L, I can solve this equation for the separation between the separation distance between planes in that crystallographic system. The next slide, I just go through some of the, found some nice pictures on the web that indicate different crystallographic planes. And so I give the Miller indices, and then I shade in the crystallographic plane to see how they're, so you can see how they're oriented, right? If you have a cubic system, right, the planes on the surface of the cube are all equivalent, right? That equivalence is indicated by putting the Miller indices inside of these curly brackets. So whenever you see 3 Miller indices inside of curly brackets, that's basically referring to all the planes that are equivalent to one another. So in this example, this blue-colored plane, this orange-colored plane, and this purple-colored plane, they're all 100 planes, and that's indicated by the notation like so. So you can start to read the scientific literature if you know this Miller index notation. I just worked through an example for a cubic system. Just ask what's the difference between two of these planes. These are 110 planes, and I just used the formula that I wrote down earlier. And if I know that the unit cell has got a dimension A0, then I can just work through this in a very straightforward fashion and calculate very quickly that the separation between these planes is A0 divided by square root of 2, which is probably a number that you would have anticipated knowing the geometry of cubes. But the point is, as HK and L get higher, right, get more complicated, this formula still works, and you can start to calculate distances between planes in a very simple way. And then lastly, I just wanted to mention that the directions inside of a crystallographic system are indicated by Miller indices surrounded by these square brackets. So here I just indicate different types of, different directions for different crystallographic directions. These directions are always perpendicular to the plane that has the same Miller indices. So for instance, this 102 direction is perpendicular to a plane that has the 102 Miller indices, right? And this just tries to bring that home, bring that point clear. So the important thing to remember is square brackets indicate directions, curved brackets indicate planes. If you remember that, and you remember that the Miller indices are the smallest integer values that are proportional to the intercepts of the plane with respect to the x, y, and z axis, you can pretty much work your way through a lot of these problems in a very straightforward way. It's just algebra, basically. So this is an introduction to the crystal systems that have been identified and how to specify planes and directions inside those crystallographic systems. And we're going to start to use this notation in some of the future lectures. And in particular, the next lecture, we're going to focus on a discussion of the energy states that are possible for electrons when you place those electrons in a periodic structure. So we want to develop a general framework to understand what happens to the electrons that are attached to the atoms, or that are associated with the atoms, when you start to pile all these atoms together to form a crystalline solid. So please come back and listen to that lecture, because we're going to start to explain to you the nature of electron states in solids. So thanks for listening to this one, and we'll see you next lecture.